We think of cancer treatment as a series of external attacks, radiation beams and chemical agents designed to poison malignant cells. Yet within our own bodies, we carry a biological defense system capable of recognizing and destroying these cells. For decades, the mystery was why this system remained passive as tumors expanded, as if it were being actively suppressed from within. James Allison grew up in Alice, Texas, a small town where his father worked as a country doctor. Riding along on house calls, Allison observed the practicalities of medicine and infection, forming an early curiosity about how the body handles disease in its most basic form. That curiosity was sharpened by loss. He was 11 years old when his mother died of lymphoma, followed shortly by two of his uncles. Allison watched the standard oncology of the 1960s take its toll, not just through the disease, but through the therapies. He saw the gaunt bodies and the deep radiation burns left behind by a medical model that relied on scorched earth tactics. These experiences left him skeptical of a system that only knew how to attack the tumor directly. He began to suspect that a more precise biological intervention might exist, one that focused on the body's internal regulation rather than external destruction. At the University of Texas at Austin, Allison moved away from the clinical path of a physician to pursue basic biological research. In the lab, he found the freedom to formulate hypotheses and test the fundamental mechanics of how life functions at a cellular level. He became fascinated by T cells, the primary defenders of the immune system. These cells are designed to patrol the body, scanning for invaders and eliminating anything that shouldn't be there. This led to a central question. If T cells are evolved to detect abnormalities, why do they fail to engage a growing tumor? The cells seemed capable of the task, yet they remained idle while the disease progressed. Allison's focus shifted toward immune regulation. He wasn't looking for a miracle cure. He was trying to understand the signals that tell a T cell when to fight and when to stop. This departure from tumor-centric research would eventually challenge the prevailing models of the era. For a T cell to attack, it needs a signal to start. Researchers knew that a receptor called CD28 acted as the accelerator, the biological gas pedal that jumpstarts the immune response. When a similar molecule called CTLA4 was discovered on the surface of these same cells, most immunologists assumed it was simply another accelerator, a second signal to further boost the immune response. Allison's research proved the opposite. He demonstrated that CTLA4 functioned as a highly effective biological brake. Once a T cell is activated, CTLA4 moves to the surface to shut the response down, a necessary check that prevents the immune system from attacking healthy tissue. This mechanism suggested how tumors might survive. As T cells move to engage a cancer cell, the deployment of CTLA4 deactivates them before they can eliminate the threat. The immune response is neutralized by its own internal off switch. Allison's hypothesis was to ignore the tumor and focus on the T cell. He proposed using an antibody to block CTLA4, effectively disabling the brake and allowing the immune system to continue its attack for an extended period. This shifted the strategy from targeting the disease to targeting the host's own regulatory system. By removing the restraints on the immune response, the body could sustain the fight on its own. In December of 1994, Allison tested this approach in mice with established tumors. The results showed that when the CTLA4 brake was blocked, the T cells eliminated the cancer entirely, and the mice developed a lasting immunity to the disease. Despite these results, the pharmaceutical industry was hesitant. Immunotherapy had a history of clinical failures, and companies were reluctant to invest in the drug that didn't attack the tumor directly. It took years of persistence before a company would agree to move the concept into human trials. During early trials of the drug, now called ipilimumab, clinicians observed an alarming spike in tumor volume. By traditional oncology standards, this indicated the disease was progressing and the drug was failing. This clinical phenomenon, known as pseudoprogression, occurred because massive numbers of T cells were rushing into the tumor site. The swelling seen on scans was caused by an intensive immune infiltration, which was followed by a sustained collapse of the tumor mass. In metastatic melanoma, a disease that was previously almost always fatal, this approach achieved durable responses. Roughly 20% of terminal patients remained in complete remission a decade after a single course of treatment. This breakthrough led researchers to identify other checkpoints, such as the PD-1 proteome. By blocking these additional breaks, checkpoint therapies have since been used to treat lung, kidney, and bladder cancers. In 2018, this change in oncology was validated when James Allison received the Nobel Prize in Physiology or Medicine. Immune checkpoint blockade is now considered a standard of care, standing alongside surgery, radiation, and chemotherapy as a pillar of cancer treatment. Allison's career-long commitment to basic science successfully tested the limits of immune regulation. By focusing on the mechanics of the host rather than the mutations of the tumor, he provided a new framework for treating malignant disease. At institutions like MD Anderson, the research continues. While checkpoint therapy has saved thousands of lives, many patients do not respond to it, and biological resistance remains a significant hurdle. Allison continues to investigate how these therapies can be combined to overcome that resistance. His work remains grounded in the same curiosity that drove his early research, a need to understand the biological barriers that prevent the immune system from completing its task. Science spent a century looking for external weapons to destroy cancer, but Allison's work showed us that the capacity for a sustained defense was already integrated into our biology. Human progress often comes from identifying and removing the specific obstacles that prevent a natural system from functioning as it was intended. In the case of cancer, the breakthrough was finding a way to let the immune system finish what it had already started.